Good morning, everyone. My name is Leslie Nelson Bernier, and I have the privilege of serving as the president of the UNC Health Foundation. And I'm so delighted to welcome you to session 2 of our COVID-19 Game Changer series. Welcome back to those of you who joined us for session 1. I think you're in for another real treat. If you weren't with us session 1, session 2 is going to be just fabulous too. We'll talk a little bit today about, we talked about testing in session 1. We'll talk a little bit about treatment today and some of the updates that have happened over the last 2 weeks. I wanted to start by giving just a few housekeeping notes, and they're on your screen here. And so if you want to mute or unmute, stop or start your video, those little icons are in the corner and circled, so please feel free to do that. Next slide. At the end of the session, you all have submitted some wonderful questions, and we will move into an extensive question and answer section. But if you have questions, please feel free to type them into the chat. We will pass those along to the panelists. And then the slide that you just saw, you'll see the participants and the little bar across the bottom too. So mute, stop video, participants in chat, all of those features should be at the bottom of your Zoom. Next slide. All right, and then additional Zoom guidance in case something happens and you need help. There's a member of our staff standing by at that telephone number and that email address if you have issues. Next slide. All right, and just a quick disclaimer, this webinar is being recorded. It's for general educational purposes only and does not constitute nor claim to be medical advice. Transmission of information is not intended to create a clinician-client relationship. So we'll get started. So I'm really excited to introduce my co-host for this morning, Dr. Matt Ewand. Dr. Ewand is the Chief Clinical Officer for UNC Health, and he also serves as President of UNC Physicians. In that role, he oversees our entire physician and advanced practice provider enterprise, which is about 3,000 or so strong. And in his spare time, he's a neurosurgeon and has a particular interest in brain tumors. So I'm going to turn it over to Matt, and Matt will introduce our speakers for the day, and we'll go from there. Dr. Ewand?
Thank you, Leslie. Good morning, everyone. It's a real pleasure for me to be here with you, and I'm grateful to have a minute or two to speak with you before I introduce our terrific panel, Dr. Boucher, Dr. Wohl, and Dr. Fischer. So I was born and raised in Saginaw, Michigan. My best friend Carl had an old wooden boat with a 70 horsepower engine, and we used to water ski on the Saginaw River and into the Tittabawassee River. It was dirty and we went under bridges, but it seemed like a great idea at the time. So 2 weeks ago, when you saw in the news that 2 dams had failed in central Michigan, I thought back to those rivers that I had been water skiing on when I was growing up. I tuned in to watch the coverage, as I'm sure many of you did, and what I saw was in the middle of a global pandemic, a community dealing with a once in every 500-year flood. And at first I looked at the houses floating down the rivers, or the river or the lake that was upstream that was totally empty with boats sitting on the ground. But quickly my eye was drawn to the people standing up on the dam. You know, in a flood like this, the people who evacuate quickly save lives because they get out of harm's way and they make sure that later rescuers won't have to come save them. And in a pandemic like we've been dealing with, the people who have practiced social distancing, even though it's put their businesses and their families at some risk or peril, have saved lives. But then there are the folks who stood with masks on top of the dam trying to keep the water from spilling over. And those folks, I think, should get special commendation. And likewise, in this pandemic, we've had a group of folks from our 33,000 people at UNC who've run towards the fight. And I'm particularly proud of those folks. And today we have 3 of them joining with you to tell you about their experience in fighting this COVID pandemic and in treating patients with that. As I said, I've never been more proud of our School of Medicine or our healthcare system, both for the care that we're providing right now as we speak, and I think Dr. Fischer is actually calling us from the medical intensive care unit, but also in the research that's being done to try to find an answer for this. So I think you're really going to be in for a treat. Let me start by introducing our panel for today. We have 3 panelists. The first is Dr. Rick Boucher. He's an adult pulmonary medicine physician, and he's been at UNC for 43 years. studying the pathogenesis of cystic fibrosis and lung disease. He's the director of the Marsico Lung Institute and the UNC Cystic Fibrosis Research Center. And he's been studying the normal physiology of pulmonary surface lung homeostasis and how those systems fail in diseases like CF. He's a national leader in that field and is working very hard now with his team to map the entry sites where the COVID virus or the SARS-CoV-2 virus enters the respiratory tract and how drugs might be used to exploit that. And then he's also the James Meeser Eminent Professor of Medicine and co-director of the Gene Therapy Center. So, wow, what a great career and what a terrific person to have on our team leading this fight. The second person is Dr. William Fischer, Billy Fischer. Billy's an associate professor in the Division of Pulmonary Diseases and Critical Care Medicine, and his research is focused on severe viral infections, including Ebola and Lassa fever, as well as acute respiratory distress syndrome. So exactly what we're dealing with now. He was one of the first physicians on the ground in Guinea, West Africa, caring for people when they had an Ebola outbreak. He now has been working closely with the WHO. And the first time I spoke to him, he was on a bullet train in South Korea, moving between sites to study and assist with how the people in South Korea were dealing with COVID infection. He's now brought those skills back to us and helped us institute them across our healthcare system and across the state. He spent his entire life fighting poverty and learned a great deal about the glaring disparities that we see across the world growing up in China with his family. And then third is our terrific colleague, Dr. David Wohl, who's a professor of medicine in the Division of Infectious Disease. And he's been using his knowledge of viral infections, epidemiology, and critical care to help track COVID patients in our hospital and throughout our community and help decrease the spread of COVID-19. He has also been a leader in our respiratory diagnostic units, which you're going to hear more about. These are our frontline ambulatory sites where folks who either need to be tested or have pulmonary symptoms come in, separating them from our other patient population. And he and his team are helping to lead the AIDS clinical trial group in a randomized trial looking at hydrochloroquine and azithromycin for early treatment of COVID. Since 2014, he's been conducting research in Liberia, West Africa to better understand treatment for Ebola, and has also studied Lassa fever. So another expert who I think will be offer really interesting insight to our group today. And just a final fun thing about David, has a passion for Italian food. So if he does well, we're going to send him a big bowl of pasta. So thank you all for doing that. We thought we'd ask each of our panelists to take a couple of minutes and give us a personal reflection on what they've been seeing and dealing with, and then each of them has a brief presentation. So Rick, I think you're first.
Yeah, thank you, Matt. For those of you who aren't in the field, quote, airway surface liquid is a nice way of saying mucus. And we study a lot of that. It's an area that at times is inelegant. People want to cough it up and get rid of it, but in reality, it's the key to protecting your lungs against the outside world. I come at it, as from Matt said, by a CF, cystic fibrosis point of view. And just to orient you, the next day you're going to inhale maybe 10,000 liters of air as you breathe normally. There's probably about 1,000 to 10,000 microbes in each liter of air that you inhale. But despite the fact then you're going to inhale somewhere between 10 and 100 million bacteria and viruses the next day, you won't have a lung infection and you won't have a nasal infection. And so we come at it from the point of view of how cystic fibrosis degrades the normal host responses or barriers, if you like, to chronic infection. And so it's a bacterial disease. So I come at it from a bacterial point of view. Unfortunately, our CF kids get what we call exacerbations. They get periods when they're much worse. And those are actually triggered by viral infections, respiratory viral infections. And typically, they start out in the nose, as probably most of you have had colds, as a cold, and then a fraction will, quote, settle in the chest. And if it settles in a CF chest, that creates a lot of mayhem for us, and we don't like that. So we've been studying that pretty carefully. And when COVID-19 came out, which has also got a second, more elegant name, perhaps, SARS-CoV-2, it looked like to us from the outside it might obey the same principles of starting in the nose and settling in the chest, but we didn't know that. We happen to have a lot of lung specimens that we use for mapping different proteins in the lung, and in our world, usually cystic fibrosis proteins. But I knew Ralph Barrick. I mean, he's a very, very wonderful, famous person who's been passionate about this type of disease for 30, 35 years, certainly as long as I've been here. And so I did call Ralph and said, look, you know, we have some reagents that may help you map the entry point for this virus. Particularly, this virus is a lock and key type of virus that has a key that is a series of little spike proteins on the surface of the virus, and it looks like a crown. If you look at the virus under a very high power microscope, and hence the name coronavirus. And so that key fits in a lock, and it's a protein receptor in the lung. And so we essentially helped him map those. And so at that point, Ralph and I started a collaboration that started from nothing 10 weeks ago to a very productive and interesting go the last 10 weeks. It's been the most passionate 10 weeks of my life, I think. So with that, I'll stop there. I'm the basic guy here, and I'll turn it over to, I think, Billy Fischer, who will give you his reflections, which are quite different.
Great, thanks, Rick, and thanks, Leslie and Matt, for the invitation to be here. It's an honor to be with everyone today. David and Rick are the real stars here, and so my job is really to highlight a lot of how important the work they do really is. So I'm a pulmonary critical care physician that grew up always wanting to be an infectious disease doctor. And so I'm really a close link between the lungs that Rick is the world expert in, and the offending viral pathogen that David has built a career on. And to tie this all together, I did all my training at Johns Hopkins and had to walk by Matt Ewan's picture every day while I was on service. So I can really unite all of the speakers today in a fun way. But because I'm interested in pulmonary and critical care, and because I have a focus on severe viral infections, I spend most of my time actually dealing with outbreaks of severe viral infections. And that includes Ebola virus disease in West Africa, as well as Democratic Republic of Congo, Lassa fever in West Africa, as well as COVID in Korea, Azerbaijan, and across Europe. And so for me, this has been an unusual outbreak in that I'm at home, but it's been important because when you deploy into these areas where these outbreaks are happening, you never fully understand the effect of that outbreak on the community and the population at risk. And so being on the other end of that really has opened my eyes to a couple of things, and I'll just share a couple of reflections on those. Number one is that everyone else's job is much harder than I had ever anticipated. And so what I mean by that is, when we created a COVID unit in our intensive care unit, we really had to restrict the number of people that were in there. And so you spend a lot of time with a smaller number of people, and you really have insight into how hard people work and how important the work that they do is to allowing you to practice at the top of your license. And I'm talking about Marta and Victor, who are from environmental services and who are responsible for decontaminating the space and making sure that we never come into contact with the virus in the first place. And they are critical to allowing us to do what we do. We initially started with a different nursing ratio. And so having to participate in moving patients and cleaning patients and proning patients, meaning turning them over onto their stomach, you gain a really true appreciation for the heroic work that the nurses at UNC do. And so for me, that's been incredibly important to see that angle. And then the other thing is that there are always ebbs and troughs, ebbs and flows to any outbreak. And sometimes we are caught up in the wave of excitement about a new drug or a new intervention. And sometimes we're depressed by the negative findings that we may see in clinical trials. And one of the things that I've learned over the years that I think is especially important today is that we have to keep an even keel, and we have to remember to do the basic things well. And if we do those basic things well, we really do put people in the best position to do well with this and other emerging viral pathogens. So I'll turn it over to David, who is also my clinical research mentor and my clinical research partner. Thanks.
Thanks, Billy, and thanks, Rick. So yeah, I think reflecting is something that we don't do enough of, especially when we're in the middle of a crisis. So I think the opportunity to do this has been useful and therapeutic even for me. So I am not normally a COVID doctor. There were no such thing as COVID doctors. And before Ebola came around, there was no such thing as Ebola doctors. And over 25 years ago, when I was deciding what I wanted to do with my life, there was no such thing really as HIV doctors. And I've realized now that everything that's come before has really led to this moment, and that I responded with many others to a pandemic 2 decades or more ago that was spreading across the world, and felt drawn to do that for a number of different reasons. I also felt it was a field that rightly would never become obsolete. There are always going to be pathogens out there, and they're always going to find a way into us. What I didn't realize is that that there would be new pandemics, and that the things that I learned first from my mentors here, learning about HIV, learning about many of the aspects of the HIV epidemic that are very operative today, and then working alongside Billy and working in West Africa on a very new, incredibly large outbreak of Ebola, really set the stage for how we responded 8 weeks ago. And now, you know, I am an HIV doctor. I feel like I'm a kind of backseat Ebola doctor, certainly an Ebola researcher with Billy, but also now a COVID doctor and a COVID specialist alongside many of us. And that's been a road that has been tricky, but it led to it. And I think Billy and I and our experience with dealing with a really horrible outbreak of a horrendous disease really helped prepare us and help point the way to others here. So I think I'm grateful for the experiences that I've had in dealing with these other really big crises that have helped us here. And like Billy said, I didn't expect it to be here again, that we'd be working on putting out the fires, but this is where we are. And last thing I'll say is, I've also reflected, and this is true, it's not gratuitous, that I feel very fortunate to be working in a system where everyone is running towards the fire, and where the institution has really almost every time said, what can we do to help? What do you need? And that has been tremendous, because Billy and I could come up with a long list of things we need, and some of them cost a lot of money. And people have been saying, you got it. If it will help, let's do it. And so that's been tremendous. And we'll highlight some of that, I think, today. So let me kick some of our discussion off, and each of us are going to talk for a few minutes, but just for next 5 minutes, just kind of set the stage and then get into some of the details. People ask all the time, and already in the question and answer, we've seen some of this, have we flattened the curve? And I figured the best way to start is by showing some curves. And you can see visually for yourself, we really haven't flattened and gone on the other side of the curves. We still are seeing increases in whatever metric you're looking at in the United States, in North Carolina, UNC Health, and at the medical center. So we are still seeing new cases. This is complicated. Sometimes we can get faked out. If there's more testing, there's going to be more people detected. But overall, we can see that there has been gradual increases. It is leveling off, but we haven't, I would say, peaked. And some of our projections that Dr. Ewan and others look at every day indicate that we haven't peaked and that we still can expect to see some new cases. Next slide, please. What we look at every day are dashboards like this. You know, we are data driven. And so we can see in real time, you know, how many people we've tested, how many people are in the hospital at any one time. Are they in the ICU? How many ventilators do we have? this is something that really, it requires considerable attention, a lot of people power to populate these figures. So a lot of data, and decisions are made on these data. And you can see for yourself some, again, the curves that we're seeing and some of the data that we're using to make decisions. I think, again, some of our projections weeks ago were much more dire than what we're seeing now, indicating that a lot of the measures that we took as a hospital, as a medical system, and as a state and as a country really have started to pay off. And I think there's really good proof of that in some of what we're seeing, and what we see in places that haven't been able to do that, where we do still see some spread, where social distancing is difficult, if not impossible. Next slide. So really, there's three aspects, I think, that stand out for me over the last few weeks of our response here at UNC, and that's detecting, identifying new cases, providing care for those who require it, and then learning from what we're seeing. Next slide. As Dr. Ewan said, we realized right away that we have to do a good job and be at the forefront in the state of identifying those who have COVID-19. And we set up a series of respiratory diagnostic centers. They're really swab clinics or swab drive-throughs where people could get tested who meet criteria. And you can see where they're represented across the state. Pretty much wherever there was a UNC affiliate or hospital, we have a respiratory diagnostic center that offers the swab deep in the nose to test for COVID-19. Next slide. So we set up one basically in a weekend. Billy was in Korea, I think, at the time. So I FaceTimed him. I said, what do you think about this? Because we've been working on Ebola treatment unit setup and Lassa fever, which is a little bit like Ebola in Liberia, West Africa, and worked really hard on developing safe zones for treating people with suspected and confirmed viral hemorrhagic fever. So really, really bad infections. And so we brought that same thinking and the same strategy, but writ large. And we took over, again, generously provided to us, a parking lot outside of our ambulatory care center here in Chapel Hill, and came up with a physical algorithm. And I won't walk you through it, but it takes people through different steps over the course of a few minutes to get them registered, find out some basic information about them, because we need to learn what we're doing and what works and what doesn't work, get them their labels, get them their baggie, and get them into the swab zone so that they could be swabbed. This whole process takes about 5 minutes. We had some folks from Chick-fil-A come down and tell us how to do this, how not to have people bunched up, and how not to have traffic going out into Manning Drive and blocking the roadway. We then set up a cough clinic in one of those trailers, A, B, and C. And C, we have a cough clinic for people who need a little bit more care. And that came online about a month ago and has been really successful. All this is not only to provide a community service, but also as a perimeter outside of our hospital. We don't need somebody with a cough who could have a respiratory virus, including COVID or other things, in a waiting room next to somebody who's immunocompromised. And this allowed us to push the perimeter of care outside the physical space of our hospitals and clinics. Next slide. So here's just some pictures. I chose intentionally rainy days because, like today, they're out there and we're out there and it gets wet, gets soggy. When we had smaller tents and they blew away, we called and we got bigger tents. And those are working fantastically well and they're much more stable. Next slide. And the other centers are doing a great job too. So what we're able to do is, track how many people are coming in, how many tests are done. So you can see the numbers here, 13,000 plus people seen. We've diagnosed almost 900 people with COVID-19. We still have some tests pending as when I did this at 7 o'clock this morning. And the numbers speak for themselves. Of course, our Chapel Hill is really efficient at doing this. Our site, people come in 5 minutes. We're doing over 200 a day nowadays, and it's really just been a tremendous learning experience and service. Next slide. And we're also able to collect data. So here are data that are pretty unique. You don't see this too often in literature, and we're getting more and more data every day. This helps us understand, well, who's being tested? And you can see it's skewing in the blue to younger people being tested, but who are we detecting COVID in? And we're seeing it's skewed more towards the bottom, more towards older people. So that's interesting, and that's important for us to understand. We could break this up in many different ways, by where people live, by race, by ethnicity, and by job. So I think there's a lot of data that we're pulling together, given the large numbers we're seeing, where we have over 200 people who've been diagnosed with COVID just at our Chapel Hill Center, and 5,000 people screened overall. Next slide. In addition, across the system, people are responding. So here's a mobile unit that Lynn Fiskus and her team have deployed in Southeast Raleigh, and that's going to other places as well. They set up mobile testing. So if people can't come to a respiratory diagnostic center, the respiratory diagnostic center is going to them. And they're screening people 3 times a week, often people who don't have access to healthcare readily. Next slide. So for care, Billy mentioned this really quickly. We devised, back of the envelope, how do we do this? How do we make zones that are safe for patients, safe for our staff? We have a dedicated ICU for people who are diagnosed with COVID, and we have now a non-ICU, non-intensive floor for people with COVID, both suspected and confirmed. We have COVID hotlines where experts can man the phones and answer questions from other physicians and clinicians in the hospital and across the system. We are administering, state-of-the-art care, whether it be convalescent plasma, and we are administering convalescent plasma as part of clinical care. Remdesivir now has become available as a non-research option, and anticoagulation are really leading and thinking about how to use anticoagulation, given clotting has been such a big issue in many of our patients. Next slide. For research, we could spend a lot of time on this, and I think my colleagues will talk more about this, but there's a bunch of different ways to think about this. And there's different therapeutic avenues, direct antivirals, monoclonal antibodies, which are really exciting, things that modulate the immune system, because we know the immune system can go a little haywire here under COVID-19, and even vaccines. And we could think about giving these to inpatients as well as outpatients, and learning very quickly that what works for severely ill people may not work for less severely ill people. And also thinking about how do we apply these tools for people who are at risk but may not be infected right now. Next slide. So there's really good examples, and each of us could talk at length about this, of where we're active right now, and not only just participating, but leading. As Matt said, there's a hydroxychloroquine azithro trial that we've launched basically today for outpatients. It has not been studied really effectively as outpatients. There's a lot of controversy about this combination. We really don't know a lot about it in people who might benefit most. Billy's been leading the study of a compound that Ralph Barrick's lab helped develop that is very exciting. There's monoclonal antibodies, one from Eli Lilly that's extremely exciting and that I think you're going to hear more about in the coming weeks. There's immunomodulators, including looking at convalescent plasma and looking at different quality or titers of antibodies within that plasma. Vaccines are going to roll out within another month or so, and that's going to be really important as well. So there's lots going on. Billy mentioned the proning study. Putting people on their belly seems to really help make a difference for a lot of our critically ill people. Billy's been a long-term champion of that, and now it's becoming really critical in most of our patients who are in the ICU. Next slide. So I'll end it there. This is Ramsey's came to visit us just last week and was a real boost to our staff who've been working almost every day for the last 8 weeks at the swab event. And again, I just really am proud of my colleagues on this call, and also just the institution for really stepping up at a time of need.
Thank you, David, very much. I'm glad you didn't show the picture of Ramsey getting swabbed. We appreciate that. So Rick, I think you're up next.
All righty, well, thanks. It's called a translational researcher. I do see patients, but I like to know mechanisms. So I spend a lot of time in the lab trying to understand how a disease evolves. Because if you know that, you can take better guesses on what to do when you're confronted with something novel like we are now. So as I mentioned, we started out by asking, which side of the respiratory tract might be first infected by this virus. And there had been a history on the predecessor of this virus, which was called SARS-1, that it was a direct lung infectious agent. And to us, even though there were not a lot of symptoms with COVID-19 and the nasal cavities, it still was not we thought it was a good bet that the nose was actually going to be the site like other viral infections. So we again had a lot of tissues, and I'll lead you through this slide, I think, quickly, but just focus on the left side, and you'll see some nice sort of purple images. The blue dots that you'll see there, the large ones, relatively large ones, are the nuclei of cells. And then you'll see labeling called nasal epithelium, which is obviously your nose, trachea, your main windpipe, if you like, in your lung, something called bronchioles, smaller airways, and then the deep lung tissue that exchanges gas is called the alveoli. So the idea is, you breathe in and your airways branch 25 times and you get gas to the alveoli to exchange oxygen and carbon dioxide. As you might expect, most of the bacteria and stuff that we talked about you inhale are impacted in the upper airways. So the lining cells of those regions tend to be taller and more robust than the lower lung cells. So you'll see in the nasal epithelium, those cells are taller than if you look down at the bottom, which are the alveolar cells, which tend to be very thin and paper-like. So that's part one. Part two then is, where is this lock that the COVID-19 essentially interfaces with in the lung? So what is that receptor, if you like? And there are two of them that sort of work in tandem, and they're in the red there, ACE2 and something called TMPRSS2. So those are the receptors that the virus locks onto to infect cells. And very briefly, if you look at those purple dots in the nasal epithelium, both on the left and on the right, you see a lot of purple dots in the nasal epithelium, and not so many as you go deeper into the lung. So the notion is that the target for COVID-19 really is the nose, despite the fact that you don't have a lot of sneezing and symptoms. So we have this wonderful colleague that Billy mentioned on campus named Ralph Barrick, who has devoted 30+ years of his life to this class of viruses. So with Ralph, we go to the next slide. Our job was to prepare cultures of these different regions, nasal, tracheal, bronchiolar, and alveolar airway cells from humans, and give them to Ralph. And then Ralph infected those cultures in a very high-tech quarantine environment called a BSL-3 unit. What you're doing, if you look at A, you're looking down at cultures that were infected with a COVID-19 virus, which is also known as SARS-CoV-2, that has a little green glowing protein inserted into it, so we can tell which cells have been infected. So if you look down at HNE, you see lots of little green dots. If you look at LAE, which is your large airways, your tracheal region, perhaps a few less, but when you get down into the deep lung, the very smallest airways, and those two things that are called AT2 and AT1, those are your deep lung gas exchange regions, much less infection. So like there's a stark gradient with receptors, there's a stark gradient with infectivity. So your nose is the target. For those of you that like numbers, in B below it, there's a graph describing how many viruses you actually get. And if you look on the y-axis of that plot, there's 10 to the 7th. That means 10 million viruses are produced within 2 to 3 days in the nasal epithelium, and you can only produce maybe 1,000 or so in the alveolar region. So a really large difference. So the nose is the engine of this infection. Now, we like to be more precise, and the nose has different cells that secrete mucus, and cells that have little hairs on them that move the mucus. So if something is trapped in mucus, it's cleared. In this case, in the nose to the back of your throat, and the same thing for your lungs, actually. So we like to know which cell type is infected, because we're scientists. We like to know at the most precise level what's going on. Next slide, please. So this is where also science can be beautiful, and as well as revealing. And so there we go. So on the left side, you're looking down, just like I showed you before, at one of these cultures. This happens to be nasal again. The green color denotes the virus and what cells the virus is in. The yellow denotes the mucus cells. And so we can then take sections through a tissue like this, much like if you were in a CT scan, and that's shown on the right. And if you look just very simply on the right, that top image, you see in the green, the cells have a little fuzzy sort of grayish lattice lace on the top of them. Those are the cilia, the little things that beat and move mucus. They're like oars that move mucus. These are called ciliated cells. So this virus likes that cell type. If you look one panel down, you'll see in the middle of that panel, maybe 3 or 4 yellow cells, and those are the mucus cells. This virus doesn't particularly care about them. So it hones in on one cell type. That's pretty smart, because if it stops the little oars from moving mucus out of the lung, the mucus will stay there, and the virus, which will be trapped in the mucus, will stay as well. So it's a way of propagating the infection. Next slide, please. Now, there's a couple of things about, we all know certain people respond to COVID one way and certain people respond to another way. And part of that response is how variable your upper airway symptoms are. You can be asymptomatic and transmit disease, or you can be symptomatic, you know, lose your smell, lose your taste, or whatever. And then a fraction get lung disease. So this is a study, again, we did with Ralph Barrick, and we took cells from 5 different people, same region. These would be essentially large airway cells, and exposed them to the same virus for 3 days. And if you look, donor 1 has got a lot of infection, a lot of green dots there. Donor 2, rather sparse. And donors 3, 4, and 5 are in the middle. So what creates that variability? Why are some people more sensitive and others are not? Now, fortunately, from the history of our center, we have 1,000 of these samples that we can test. So there'll be genetic components. Certain people will have more of the receptors. Certain people will have more genes that help fight off viruses. And others have other intercedent or complicating factors, and particularly asthma. So I think with this kind of research, we can get a feel for why somebody's more sensitive than another. And then finally, a fraction of folks get pneumonia. And a viral pneumonia with COVID is a bad thing, as Billy has to deal with every day. So the next slide is, how does a virus settle into your chest? And so how do you get pneumonia from something that starts in your nose? The short story is that the virus comes from your nose, collects in your throat, and unfortunately every night we all aspirate a little bit of saliva. You know, our plumbing is not perfect, and a little dribble of fluid goes down into your lung. So, and that panel shown as A, on the left side, the far left side has got a small eye under it. That's just a stain of a lung of a COVID-19 patient. This patient actually came from New Mexico. The scientific community is amazing and be able to transmitting samples around the country and collaborating. The second panel over, it looks very pale. That's just a control. The third panel over that you see SARS-CoV-2 at the top in red. You'll see on the right hand, lower right hand panel there's a lot of red there, and that's actually the stain of the virus. And if you look around it looks pretty good. So what happened is a small, essentially droplet from your throat was inhaled at night down an airway into this section of the lung and infected it. So there's something in our world called oral lung axis, and this is what it is. Things that are in the throat, unfortunately, get down into your lung. So part of the variability then in the pneumonia is how much you or I or other folks aspirate at night and how much virus is in there at that particular point in time. And just to finish it up, in the panel B there, we always like to know if our cell culture model systems are accurate. Do they predict what's going on in vivo? And maybe if you just looked at the panel with the 2 small eyes on the right and B, you'll see in orange there, that is COVID virus. And right above that orange in most of those cells, you'll see that greenish material, and that is indeed those cilial hairs. So in the lung of COVID-19 patients, again, it's that ciliated cell that selectively gets infected. So I think just to summarize, from my point of view, you know, this virus is a highly contagious virus. I think it's essentially, it's It's generated when you and I talk. There's a great study showing how certain consonants, the TH that we use, the will produce lots of particles. Those particles are filtered out of the nose first. The nose, next 3 or 4 days, gets the virus. And then if you're unlucky, you aspirate it at night and get the mnemonic phase of this. So we privately, from the CF world, we've faced this for years. So we're big fans of, nasal hygiene. So we're big fans of neti pots. We're big fans of putting a little bit of Johnson shampoo, frankly, into a neti pot to clean things out, which will actually kill this virus. But as I'm sure Billy and David will emphasize, we're huge fans of masks all the time, because this thing is transmitted by normal tidal breathing and normal tidal talking, and it goes straight to your nose. And we are big fans of social distancing, because these particles, will not make it in a normal environment past 6 to 10 feet. So I think I will stop there and turn it over to Billy.
Great, thanks, Rick. And I'm glad these two guys went first, because what they're talking about is really exciting. Because what you've heard about is really the two extremes of a spectrum that really exemplifies UNC's purpose-driven response. It's both the bench and the bedside. And in any outbreak, one of the most important aspects of response is identifying that infected patient. If you can identify that infected patient and isolate, then you can prevent ongoing transmission from that person to other people. But it also affords you an opportunity to intervene with both supportive care and potentially an efficacious therapeutic. And so that's why the RDC and the work by David and Melissa Miller and others is so important in terms of identifying that infected patient. But once that person's identified, their body really represents a battlefield between the virus and the host. And if you think about this battle, there really are no other people. The two world's experts who can represent both sides are really Rick Boucher, who is the world's expert in the lung and respiratory epithelium, and then Ralph Barrick, both at UNC. And Ralph Barrick is one of the world's leading virologists for coronaviruses. And there have been many of us that have talked about the threat and potential of a coronavirus pandemic for years. And Ralph's actually been doing something about it while everyone's been talking about it. And over the past years, Ralph and his lab have actually been screening thousands of compounds, looking for anything that's efficacious against the coronavirus before they're needed. And so they actually cooperated and partnered with some drug companies, including Gilead, to screen thousands of compounds in these rare culture systems to try to identify effective drugs. And one of the drugs that they identified was remdesivir. And in large part, the reason that we have effective therapies today against this virus is due in large part to the work that Ralph's lab has done. But remdesivir's got an interesting and a circuitous history. The drug actually also showed efficacy against another virus that we studied, Ebola virus. Nobody likes to study that virus as much, but this is something that David and I have spent a lot of time thinking about and working on, primarily because it affects vulnerable populations in West and Central Africa. And so this compound actually looked like it demonstrated really good efficacy in both cell culture models as well as non-human primate or monkey models of Ebola virus disease. And so about 2 years ago, I was deployed by the World Health Organization into the Democratic Republic of Congo, into the eastern part of the country where there's active conflict. Essentially, there's a war. And the appeal of going, it's difficult to explain, but we had the opportunity, the first time ever, really to introduce novel therapeutics into this outbreak. And so it was a chance to really fight back, to push back against that virus. And although remdesivir didn't turn out to be the most efficacious drug against Ebola, we learned a lot about how to deliver it. And we learned a lot about its safety and tolerability. And that information, that experience, combined with Ralph's data and Ralph's work demonstrating how good this drug looks against coronaviruses, really allowed us to respond very quickly in this outbreak. Within 5 days of the announcement of this outbreak, Ralph was on the phone advocating for the consideration of remdesivir as a therapeutic in a clinical trial. And the Chinese and across the world were able to launch a clinical trial remdesivir very quickly. And one of those trials has just resulted on Friday, actually, which demonstrated a significant decrease in the duration of illness and almost a significant decrease in mortality. And so if you think about this, this is really a big deal. Speed can be counted in terms of days, in terms of weeks, in terms of months, and unfortunately in some outbreaks in years, but it can also be counted in terms of lives. And the work that Ralph was able to do before the outbreak happened really allowed us to get a drug that looks very promising into use very quickly. And so I think that can't be understated enough. And the good news is that Ralph hasn't stopped working. He is in his lab with Tim Sheehan, and those guys have been screening other compounds, and they've recently identified another compound that looks really promising as well. So even though remdesivir looks like it's working pretty well, they have a new compound called NHC, and this drug looked good in cell culture models of coronavirus, of SARS-CoV-2, the virus that's causing the COVID-19 pandemic. And it also looked good at protecting mice that were infected with other strains of pandemic potential coronaviruses. And so based on that experience, we reached out to the company that's currently working with it. We said, hey, why don't we continue this pipeline? Why don't we extend the pipeline from Ralph's work into people? And so we are about to actually launch a phase 2 study looking at the antiviral efficacy of NHC, as well as safety and tolerability in outpatients that are infected with this virus. So this is really exciting to continue to do this work at UNC. And what's really exciting about this therapeutic is that it's an oral drug. So this is a drug that could potentially be used in outpatient populations, and that's the population that we'll be studying. To try to see if we can decrease both progression of illness as well as potentially replication of the virus. So extending this development pipeline right here at UNC, I think is incredibly promising. So what does all this mean? We've talked to you about ciliated cells. We've talked to you about swabs that go in an uncomfortable place in the back of your throat. And we've talked about new drugs that are emerging and look promising. And I think what this means to me, and I'm reminded of this every day, is that it means that when somebody is sick, when a 25-year-old woman who's pregnant and in respiratory failure shows up to the intensive care unit, it means that we can tell her that she came to the right place, that we've got her, that we can take care of her. And even though she has to go on life support, which is incredibly complicated for anybody, let alone somebody with a high consequence pathogen and who happens to be pregnant. It also means that we can take care of the most complicated patients, even two patients at the same time. And this particular woman did come to UNC, and she did receive incredible care. And she did actually receive remdesivir. She was one of the first people that we treated with this drug. And I was actually the attending on when we started the drug. And as I was infusing it into her, I was reminded of how far this drug had come. But I was also reminded of the fact that it really just came from the building next door, from Ralph's lab to safety and tolerability in our hands, and then finally into this woman's arm. And you know, it's amazing when you think about all that went into saving her life and getting her off the ventilator and discharging her with both mom and baby doing well. I think it speaks volumes, not only about our ability to respond now to the patients who need care now, but also in terms of the platform that's been developed at UNC to make sure that we continue to develop effective therapeutics, effective clinical interventions, so that we can continue to address the needs of the state and beyond. And I'll stop there, turn it over to Matt and Leslie.
Thank you, Dr. Fischer, Dr. Wohl, and Dr. Boucher. This was just such a fascinating and interesting set of presentations, and amazing to see the terrific work that's happening across the UNC Health system. As you know, we have a number of questions that have been submitted prior to the meeting today, as well as some that are coming in on the chat. So please feel free to keep sending them in. We'll get to as many questions as we possibly can in the next few minutes. But I'm going to start with Dr. Wohl and ask someone wants to know what you think about flying, staying at a hotel, or camping. Would you do it right now?
Well, I like all those things, but I don't like them right now. And I think, so much of what we think about in infectious diseases is what's my risk? So my risk tolerance and Billy's risk tolerance when we fly over to Liberia during Ebola time may be different than somebody else's risk tolerance. So there's individual decision making here, but our decisions have consequences. So right now, you saw the curves. We are not at the bottom of the mountain. We're still at the peak. And I would be really concerned about being too close to other people. My door is shut. I think every time Dr. Ewan's door gets open, he puts on a mask, I've noticed. So, he's got it, I've got it. There's a reason we have these things. I don't want COVID. If you don't want COVID, you have to stay pretty far away from other human beings.
Okay, thank you, Dr. Wohl. Dr. Boucher, this next question is for you. So you are an expert in cystic fibrosis and bacterial infections in particular, and you've dedicated your whole career to this. What led you to reach out to Ralph Barrick, who's in our School of Global Public Health, to start working with him? And does it speak to the collaborative nature of research here at UNC?
Yeah, I'll be quick because I sort of got into that a little bit in my opening preamble. But again, I deal with a group of patients that get, unfortunately, intermittently hammered by viruses. So we've had to deal with that problem. And so we've been looking at it with the more common viruses that we all get, which are called common cold viruses, rhinoviruses, RSV, or the croup virus. But this one, when we looked at it, we thought had the same principles. It was just a pleasure. I'd known Ralph before, and I had known what a fine quality scientist he is and a fine human being. So it was easy to set up a quick call, 10 minutes, make a deal. He did this, we did that, and after that, the calls unfortunately got longer and longer as we got more and more into it. But it's been a total pleasure. And again, for those of you who may not know the structure of UNC, Ralph is officially in public health, but he's co-appointed in the School of Medicine, and a lot of our collaborators are in pharmacy. So it's in fact, we have a lot of mathematical modelers up on campus and physicists, to be truthful. So it's a very collaborative environment, and it's just a total pleasure to work in it. So I'll stop there.
Thanks, Dr. Boucher. Dr. Fischer, you've been on the front lines fighting Ebola in Africa. How do under-resourced, underserved communities and populations combat this disease if we're having such a hard time here just in our country?
Yeah, I think that's a really important question. It's one that we've been thinking a lot about, and it's going to take a global effort to support these under-resourced areas. For a moment, I would argue that the U.S. was under-resourced. We didn't have enough testing capabilities. We didn't have enough swabs, and so in some places we didn't have enough ventilators. And so I think we experienced a little bit of what that's like. The most important thing that we can do as a global community, because these diseases are global threats, is to respond together. We have to identify places where there's outbreaks and we have to go to the source of the outbreak. If we do this every single time, if we respond to the source of the outbreak and put it out at its source, not only are those populations better off, but actually the rest of the globe is better protected. This is a key piece. And this is one that has not been embraced globally. But I think that hopefully that this experience will change that.
Great, thank you. And Dr. Ewan, there's a question here for you. Will there be a second wave, and are we better prepared to face the consequences of it?
I think I'm probably the least qualified to answer this, but I'll take a stab at it. Winston Churchill said, "This is not the end. It is not the beginning of the end, but it is perhaps the end of the beginning." And so I think when we first faced this in March, we had the same mentality that North Carolinians have for a hurricane. When will it be here? When will it be gone? And how bad will it be? And I think what we're seeing is that this is playing out in North Carolina at a slower pace than we initially anticipated, which does not mean we should take our eye off it, but it does mean we've had more time to prepare for Dr. Wohl and Dr. Fischer to prepare our treatment strategies, for us to procure masks and other personal protective equipment. So I think we'll be better prepared because we're smarter and we've had more time, and our population is more well educated about the things they need to do. But I think there's still formidable work to be done, and just grateful to have such a great team to do it with.
Thank you, Dr. Ewan. So I know there are a number of additional questions that we didn't have time to get to today. We will definitely reach out to you if you're interested in hearing from us one-on-one to answer some of those questions, and we'll keep them for session 3. So I'm going to close this out today just by pointing to a few things for you, but our resource, UNC Health's COVID information that you can go to find out more about what we're doing in this space. And if you've been moved by any part of what you've heard today, I hope you'll consider making a gift to our COVID-19 Response Fund. I'd also ask that you think about others in your network that might need to hear this story and connect us with them. There'll be a video of this session for you that we'll send out in the follow-up materials so that you can forward it on to your network or friends or colleagues or family. And then lastly, I just want to put in a quick plug for part 3. So session 3 of our COVID-19 Game Changers will take place on June 10th. And that will again be from 11 to 12. And we will welcome 3 additional specialists that are helping us deal with COVID-19. So I guess almost lastly, I'll say that there is a post-event survey that's going to come up on your screen. If you'll take a couple of minutes just to fill that out, that would be great. And again, thank you so much to my co-hosts, Dr. Ewan, Dr. Wohl, Dr. Fischer, and Dr. Boucher, and especially to all of you all. Thank you very much for being with us, and we'll see you next time. Bye.